But before making the biodiesel through the process of transestrification, we must first go through what is called a titration. You might be asking yourself, what is a titration? A titration is basically just a chemical process to determine the concentration of a solution. It involves two solutions, one with a known concentration that is called the titrant, and one with a known volume but an unknown concentration called the analyte. As you will see during her lab, Emma performed what is called an acid-base titration, in which her titrant, a strong base, was a concentration of 0.1% mass volume of potassium hydroxide, KOH, and her analyte was an unknown concentration of a weak acid, which were the fatty acids in the vegetable oil. The purpose of this first part of the Biodiesel Lab was to titrate two analyte samples with a known concentration of potassium hydroxide in order to determine the amount of free fatty acids, also known as FFAs, in a sample of used vegetable oil, and then with this information to figure out the amount of potassium hydroxide needed to convert as a catalyst this used vegetable oil into biodiesel. In addition, it is very important to determine a precise amount of catalyst in order to avoid an unwanted chemical reaction that occurs during the transestrification process. This chemical reaction is called a saponification. A saponification is a process in which there's a formation of soap. This occurs when there's a hydrolysis of esters with a strong base. This unwanted chemical reaction could potentially occur during the transestrification lab if the amount of potassium hydroxide isn't precisely calculated beforehand. This is because when making the catalyst, so by mixing the methanol with the potassium hydroxide together, there is a possibility for the OH and the K ions to separate from the potassium hydroxide, and then the OH ions would form water, H2O. During the transestrification, if the H2O comes in contact with the free fatty acids, this would create the soap. This is why there could be a soapification. Now let's talk about the procedures and the different materials used during the lab. First, Emma needed to assemble the titration montage and go collect about 5 milliliters of used vegetable oil in a beaker. She then filled a 50 milliliter burette with a 0.1% mass volume concentration of potassium hydroxide. She then removed the air bubbles inside it by gently tapping on it. She then washed a 20 milliliter volumetric pipette using first distilled water and then using isopropanol. She then filled 20 milliliters of isopropanol into the volumetric pipette and then poured it into the 50 milliliter beaker. She then added 1 milliliter of used vegetable oil and 2 to 3 drops of phenol red, which served as the pH indicator during the titration lab. She then added a magnetic steering peanut to the 50 milliliter beaker and placed it on the hot plate under the burette, which contained the strong base solution of potassium hydroxide. As you will see in a few seconds, the phenol red drops will enable the solution of titrant and analyte to turn pink. This indicates that the solution is now turned basic and has a pH of over 8. For her results, in order to find the average volume of KOH used during both her titrations, she measured the initial and the final volume of her potassium hydroxide. Then, with these volumes of KOH used, she plugged it into the formula percent mass volume of free fatty acids equals volume of KOH times 0.502. She then found that the average calculated percent mass volume of free fatty acids in the oil was about 1.33. She then proceeded to figuring out the amount of potassium hydroxide needed for the transestrification lab. Her result was 0.504 grams. In the end, though the titration process isn't the most crucial step in order to make biodiesel, it sure is really important when it comes to properly executing the transestrification process.